Thank you for the introduction. So I'm going to talk about computational toxicology, nanotoxicology, and in particular modeling the biokinetics and toxicity of nanomaterials. And I'd like to start with really sharing a few words of why we think computational modeling is useful in the assessment of nanomaterials. So of course we can assess the biokinetics of a substance within the body, and we can also assess the dynamics of the biological response that results once the substance has reached the target organ of interest. So of course, if we're interested in modeling the biokinetics in vivo, then we can use physiologically based kinetic modeling. If we're interested in modeling the fate of chemicals within an in vitro test system, there are also mathematical models for this purpose. And these are, of course, biologically based mathematical models, but there are also structure-based mathematical models, and in particular, quantitative structure-property relationships, which are mathematical relationships that allow you to predict physicochemical properties, and quantitative structure-activity relationships, which are models that allow you to predict biological activities such as chemical toxicity. And these models can, of course, be used in their own right, but they can also be used to provide input parameters to the more complex PBK models. So these are the kinds of models that I'll be referring to in the rest of this presentation. When we're talking about structure-activity relationship modeling, I think it's useful to think of three different kinds of approaches to this. The first of which is essentially theoretical. You're deriving the models from first principles. So for example, you might be modeling chemical reactivity based on the energies of the molecular orbitals in the substance of interest. The other approach is entirely statistical. You're essentially using pattern recognition in order to correlate descriptors that are potentially useful with the effect you're trying to predict. But most SAR modeling today, I would say, is what you might call hybrid modeling. That is a combination of mechanistic reasoning and statistical fit. So you use theoretical considerations to identify the descriptors that are likely to be predictive, but you use statistical modeling in order to calibrate the parameters surrounding those descriptors. So what are the useful physicochemical properties for predicting biological activities of nanomaterials? This is taken from a draft ECHA guidance on the grouping of nanomaterials for the assessment of these substances under REACH, the European chemicals legislation. And you can see they distinguish between three kinds of properties. The first kind of properties are essentially describing the identity, the chemical and physical identity of the substance. And then you have the more extrinsic properties that describe the fate of substances within the body or in the environment where they go, and the reactivity and toxicity of these substances. So this is a kind of typology of physicochemical descriptors, if you like. So to illustrate, again, a model, an SAR model that is based upon mechanistic reasoning, here is an example where the point of zero zeta potential or the point of zero charge is predicted from electronegativity. And the electronegativity can, of course, be looked up in a physics handbook. And in this case, the point of zero zeta potential is being used to predict the agglomeration aggregation behavior of nanomaterials. And if you generate the computed point of zero zeta potentials for a whole range of metal oxides, you can then overlay that onto the pH ranges of the different fluids within the body. And that might give you an idea of whether or not you would get agglomeration within those particular tissues. So if we're more interested in predicting chemical toxicity, there are many mechanisms of nanomaterial toxicity, as we heard in Andre Nell's presentation yesterday. One that's commonly modeled is toxicity based on chemical reactivity. And this is based on really the belief that if a nanomaterial is able to exchange electrons with the cellular environment, it can disrupt the cell redox state, and this can lead to oxidative stress. And of course, the cell has only a certain capacity to compensate for oxidative stress, and if this is too prolonged or too severe, then this can translate into adverse effects, cytotoxicity, inflammation, and other adverse effects in the body. Here's another example of using structure-activity relationship models for carbon nanoneedles, and this is based entirely on a molecular modeling study. The study is actually almost 10 years old now, but what we're modeling here is the effect of the size of the ring within the nanoneedles, whether it's 4 carbon atoms, 6 carbon atoms, or 8 carbon atoms, and the effect that the length of the needle has on the reactivity. So you can see that as the nanoneedles are actually made to be longer, the gap between the HOMO energy level and the LUMO energy level actually decreases. So in a sense, the nanoneedles become less stable, more conducting, more reactive as they become longer. So here's a simple example of a structure-activity relationship based upon mechanistic reasoning. Now, this works very well for molecules and long molecules such as carbon nanotubes, carbon nanoneedles, but it doesn't really work for chemicals in a crystalline form, because here, of course, the close proximity of the many atoms means that the molecular orbital energies tend to overlap and they split, and what you actually get is a valence band and a conduction band, and these are separated by an energy gap. Now, a former colleague of mine, Enrico Borello, hypothesized that this can be used in order to predict the potential of nanomaterials to disrupt the redox state of cells. And in particular, if chemicals in the cellular environment are able to donate electrons to the conduction band of the nanomaterial, or if they're able to receive electrons from the conduction band, then in a sense, the nanomaterial will be acting as a catalyst and potentially able to cause oxidative stress and adverse effects based upon an excess of oxidative stress. He illustrated that this was in fact the case for a number of metal oxide nanomaterials, and here you can see where the conduction band overlaps with the range of redox potentials that are observed in the cellular environment. You would predict oxidative stress and potentially cytotoxicity and other effects. And this was demonstrated both in terms of the scientific literature, and subsequently it was also confirmed experimentally by other groups, such as Andre Nail's group in the US. This is a different example. This is more of an example of a hybrid QSAR model, where the cytotoxicity to a cell line for a number of metal oxides is being modeled on the basis of two chemical descriptors, one of which is the enthalpy of formation, so that's related to the band gap energy and the stability, and the other is related to electronegativity. And both of those descriptors are computed, so it's a very simple type of QSAR model for predicting cytotoxicity. Now, in the next few slides, I want to share some of the results that were obtained in a project called Nanocomput, and this was a project internal to the European Commission that the JRC carried out on behalf of DG Growth. And the purpose of this project was to evaluate the availability and the applicability of various computational methods for the assessment of nanomaterials under REACH, the chemicals legislation. And the results I'm going to show you were actually provided under contracts by LEITAT Technological Center in Barcelona. So that's Alejandro Vilches and colleagues who did this work for us. So we're looking at the landscape of different models from a systematic review of the scientific literature. And we wanted to do this so that we can identify the kinds of nanomaterials that are being modeled, the kinds of descriptors that are being used, the statistical methods, and the endpoints that are being predicted. So you can see that this review of the scientific literature identified just over 120 models, structure-based models, most of which are QSAR models, but there are also some QSPR models. Most of the QSAR models are predicting cytotoxicity or some sort of combination of cytotoxicity endpoints, and most of the QSPR models are predicting solubility. So this actually means, for the purposes of REACH, most of these models are not actually particularly useful, because cytotoxicity is not a required endpoint under the chemicals legislation, and you would probably want to measure solubility rather than predict it. But the value of these models is rather different. The value of these models could be in terms of grouping nanomaterials based on their similarity of in terms of physicochemical properties, and similarity in terms of their biological activities. And then you can read across the toxicological properties of these nanomaterials between members of the group. So this would be the value under the REACH legislation. Turning now to the more biologically based models, we can see that not many physiologically based kinetic models are identified in the literature. In fact, just 24 such models, and these were mostly developed for the assessment of fate of metals and metal oxides. It's also important to note that these models carry with them quite a number of assumptions. For example, they assume that there's no agglomeration and aggregation within the blood. They assume that the nanomaterials are equally distributed throughout each compartment. So they make a number of assumptions that make it perhaps difficult to scale up these models to nanomedicines. Looking at the landscape of these other mathematically based models, we can see that there are really fewer than 10 models that predict in vitro dosimetry. So these are actually useful for predicting the target doses of chemicals in vitro systems and for interpreting the results of toxicological experiments in vitro. And what's interesting about these models is that they're including a number of fundamental processes such as aggregation behavior, sedimentation behavior, and advection behavior within the in vitro test system. And then, of course, there are a number of models of the respiratory tract that can be used to really simulate the fate of chemicals within the airways and within the lungs. And this is obviously of great interest in terms of occupational exposure. So the overall picture is one where we have thousands of these kinds of models for small molecules. We have a lot of experience in this respect. The application of these models to nanoparticles is still in its infancy, but progress is being made. And I would say that the development of these models for complex nano-objects such as nanomedicines is something for contemplation and future research. And with that, I'd like to come to some conclusions that essentially modeling nanomaterials is quite difficult because of the chemical and physical complexity of these substances. And of course, they have properties associated with both the solute and the particle phases. As I mentioned, agglomeration and aggregation behavior means that you don't have a single species to model as you would normally consider in traditional QSAR modeling. The properties can depend on the medium, and this can be time dependent. We've heard a lot in this conference about the protein corona. One of the challenges is that there is a lack of computable and toxicological relevant descriptors, and there is a lack of reliable in vitro and in vivo data for the model calibration. However, we found that some physicochemical properties are useful surrogates for biological activity, and in particular, chemical reactivity has emerged as a useful predictor of the toxicity of these materials. And with that, I'd like to thank you for your attention, and I leave you with a few references in case you want to follow up on some of these ideas. Thank you.
Questions? No questions. I have a question myself. The needles that you have, are they stable material?
Are they stable?
Ever been made, these needles, carbon needles?
This was an entirely theoretical study, as I mentioned, really just to illustrate the dependence of reactivity on...
They look very unstable material.
Yeah, we didn't verify that experimentally.
Okay, fine, thank you. The next speaker is Anand Priya